So in this last section on carbohydrates, we want to talk about polysaccharides. So polysaccharides are going to be polymers of tens, hundreds, or even thousands of monosaccharides that get linked together through these glycosidic bonds. So we've been looking at the dehydration reactions that can link the anomeric carbon position on one sugar molecule through one of the alcohol functional groups on another molecule. So the three most important polysaccharides are cellulose, starch, and glycogen. So all of these major polysaccharides are built up from glucose monomers, cellulose, starch, and glycogen. So the cellulose will contain beta 1,4 linkages. So it's got glucose in the beta anomeric form. So that means this first hydroxyl is going to be in the up conformation. And you can see that it's a beta 1,4 linkage that's going to link the two sugar molecules together. So you've got this one in the up conformation. The 4-hydroxyl is in the down conformation. So essentially, every other glucose molecule will be flipped over to make the cellulose structure. The starch and glycogen repeating unit is the alpha 1,4 linkage. So you can see that the alpha anomeric form of glucose has the first hydroxyl group at the anomeric carbon position going in the down conformation. So this small change in the glycosidic linkage, whether it's in the down conformation or the up conformation, completely changes the structure of the polysaccharide. So the cellulose, remember, is a very structurally sound component. It makes up things like the cell wall and modified cellulose sugar units. So if it contains a nitrogen group in there, making the N-acetylglucosamine or just the glucosamine molecule, it can make up the chitin structure that makes up the very hard exoskeletons of things like insects. So the cellulose repeating unit is very structurally sound and difficult to break down. However, the alpha 1,4 linkage is what we consider food molecules. So things like starch and glycogen. Starch is the major storage form that's found in plants, and we'll talk about the components of starch and what that polysaccharide looks like. And then glycogen is going to be the major polysaccharide storage of glucose in animals. And so we'll talk about how the starch and the glycogen differ from each other, because they do both contain the alpha 1,4 linkages. So this is giving you an idea of how the cellulose actually looks when it's forming its large polysaccharide. And so you can see that there is a flipping of each sugar molecule. So every other sugar molecule is going to be flipped upside down. So you can see the linkage up here between one, and then the next one is going to flip over so that it can attach. And you can see the linkages alternate above and below the plane. And this is important because it sets up a dynamic of hydrogen bonding between the alcohol functional groups on one sugar with the alcohol functional groups on another sugar. So you see all of these red indicators here are hydrogen bonds that end up forming between the sugar molecules that make up the cellulose. And it's this hydrogen bonding character that you get from the beta 1,4 linkages that give the chains their toughness and fiber quality that the cellulose is going to have. So you know the fibrous material that you find in plant material, like in hemp, for example, that makes good rope, is going to be composed predominantly of cellulose. So cellulose is also the major constituent then of the plant cell wall. And this is very important because plants rely a lot on water pressure to maintain their structure. And so they have a huge organelle that plant cells do not. They have a vacuole that's going to carry water. And so they actually like to stay in a turgid state. So they like to stay pretty full of water. And the cellulose cell wall is going to make it possible for them to hold a large amount of water without bursting. Animal cells do not contain a cell wall. Animal cells only have the plasma membrane, which is the very thin lipid bilayer. So the plant cells also have the lipid bilayer. It's on the inside of the cell here. It's on the inside edge of the cell. of the cell exterior. So you can see kind of this little pink line running through here. That's the plasma membrane. And then this thick green boundary here is indicating the formation of the cell wall, which is composed predominantly of the cellulose or the polysaccharide. So you've got lipid material inside, cell wall, which is the polysaccharide on the outside. So this is contrasting then the plant and the animal cells, showing that the animal cells only have the cell membrane. Again, the plant cells have both the cell membrane and this exterior cell wall, which allows them to hold a lot of water pressure. So if you put most animal cells in a water environment, they're going to fill very quickly with water and they can burst. So things like red blood cells, for example, would burst if you put them into straight water, whereas the plant cells do not. So we said that the exterior of beetles and other insects is made up of these beta linkages. So this is essentially just like the cellulose molecule that we took a look at, except the sugar residues are modified. They're modified with the amine functional groups so that they have the acetylglucosamine. So they've got this amine group and the acetate group or the acetyl group hanging off the end here. Other than that, structurally they are very similar to what we see for cellulose. And if it's combined like in the encrusted form with a salt like calcium, you can get very structurally rigid materials like you would see in this beetle's exoskeleton. If you don't have the salts or the metal component in there, it's going to end up forming a more leathery type of material like you would see in the skin of the caterpillar. So our other two major polysaccharides that we're going to take a look at, our starch and our glycogen, have the linkages within the glucose molecules in the alpha conformation. So they're going to be alpha 1,4 linkages are going to be the predominant form in starch. So within starch, there's two major types of molecules. There's the amylose, and so amylose only contains the alpha 1,4 linkages between the glucose molecules. So amylose consists of about 20% of the starch, and it contains up to 1,000 alpha D glucose units that are all connected together in one big long chain in these alpha 1,4 links. So instead of lying side by side and flat as in the cellulose, amylose will tend to form these coils and move into a helical type of structure. So this one is not really structurally sound, but it's a very tasty food molecule. So I love starch. So amylopectin makes up about 80% of starch, and it's very similar to the amylose, but it has much larger molecules, up to 100,000 glucose units per molecule. So these are very, very large. It also has an additional linkage that can happen within the glucose chain. So you still get the bulk of the linkages are between the alpha 1,4 positions. But now you can have branching. So you can have branches that extend off of the 1,6 linkages. So you can also have alpha 1,6 linkages. So essentially you've got your main chain that's going down here, and then at every branch point you're going to have another chain that's extending again in the 1,4 position here until you reach the branch point, which has this alpha 1,6 linkage. So it's linking through the hydroxyl group of the 6 carbon. So these branches happen about every 25 units along the chain. So this is a model then of the amylopectin. So each of these dots is representing a glucose molecule, and you can see that there are branch points at the 1,6 linkage about every 25 glucose residues. So this makes up about 80% of the starch molecules has the branching. About 20% has no branches and just contains the alpha 1,4 linkages. So if we compare that to our third type of major polysaccharide, the glycogen, which is the major storage form in animals, what do you notice about the glycogen? Hopefully something pops out at you. Glycogen has much more branching than the starch does, than the amylopectin. So it has branch points about every 10 glucose residues. So you get a much more branched carbohydrate storage unit. And this is very important because it takes an enzyme in the body to actually break down and hydrolyze the glycogen. So in lab this week, we actually did hydrolysis of the starch molecule, and we actually did that as a demo for you. And this was the part C. And so what we did is we put the amylose and amylopectin, or our starch mixture, into an acidic condition, and then we boiled it in acid, essentially. And when we boiled it in acid, we were able to hydrolyze all of the glycosidic bonds and break down the starch into the glucose monomers. So we can do that if we have a lot of acid present and high heat, very high heat, boiling temperatures. So in the body, that's not possible to do that. Sure, in the stomach, you have very acidic conditions, but most of our digestion of carbohydrates actually happens in the mouth and in the small intestine. And so under these conditions, the pH is pretty close to neutral, and we don't have boiling conditions inside the body anywhere. So to be able to break down these molecules inside the body and digest the starch or the glycogen, we have to have enzymes present that are going to help us mediate those reactions. And so the enzymes are going to make the reactions happen much faster because they are facilitating or bending the molecule in certain ways to allow the reaction to occur. So the enzymes that end up breaking down the glycogen in human cells only can work at the end of the glycogen molecule. So they can only cleave off this glucose at a time. If you only had one linear chain of the glucose polysaccharide present in the cell, it would take a long time to be able to release glucose to be utilized to make energy. So we utilize the glucose molecule to make ATP. And ATP is important for doing everything in the body. It's important for our brain to be able to function. It's important for muscle contraction. It's important for cell-cell signaling. It's important for almost every process inside the body. ATP is like the energy currency in the cell. So if we only could knock off one glucose molecule at a time to help us produce ATP, we would never be able to produce enough in the body. So glycogen is very highly branched, and glycogen is made and stored predominantly in the liver. And in the liver, it's going to be able to be broken down very quickly, and it's going to be able to release that glucose into the bloodstream so that you can maintain your blood glucose levels. And this will help keep maintaining your muscles and your brain in very good working order because it's going to provide the energy to do that. So by having this highly branched structure, you can get glucose molecules to start breaking off of this molecule at multiple places. So all of these can be attacked by enzymes so that you can release hundreds of glucose molecules all at the same time, rather than having to do it one at a time. So this would be very critical if you were faced in a situation where you had to run away from a lion, for example. So your fight or flight response is going to take a lot of energy. So you can have energy availability very quickly when you have lots of branches. The plants, on the other hand, they don't need to move around and run away so much. So they have much less branching because they don't need energy quite as quickly to make energy for their cells to function. They still do need some energy, and they do constantly break down their starch molecules to provide that energy as well. So those are the major polysaccharides that we're going to talk about. So we talked about cellulose with the beta-1,4 linkages. We talked about the chitin, which is very similar to the cellulose. It has the beta-1,4 linkages as well, just the modified glucose residue. And then our starch and glycogen with mainly having alpha-1,4 linkages, and also the amylopectin and the glycogen having the alpha-1,6 linkages, so that you get branching for easy release to release energy. So that is the end of our carbohydrate section. What we should have learned is that monosaccharides are compounds with 3 to 7 carbons, and either the aldehyde on the C1 position to form the aldose, or a ketone group at the C2 position to form the ketose. On all of the other carbons, you're going to have the hydroxyl or the alcohol functional groups. So remember, our formula for carbohydrates is a 1 to 2 to 1 ratio of carbons to hydrogens to oxygens. If you had a 7-carbon sugar, for example, that would mean that you'd have 14 hydrogens and 7 oxygens. It would be a C7H14O7 compound. So disaccharides, remember, consist of 2 monosaccharides, and polysaccharides are going to be large polymers of up to thousands to hundreds of thousands of monosaccharide units all linked together in glycosidic bonds. Remember, our monosaccharides can contain several chiral carbon atoms, each bonded to hydrogen, the hydroxyl group, and 2 other carbon atoms in the chain. So a monosaccharide with N chiral carbon atoms can have 2 to the N stereoisomers. So that's our formula. So if you have 4 chiral centers, for example, you would have 2 to the 4 stereoisomers, or 16. 2 times 2 is 4, times 2 is 8, times 2 is 16. So, and remember, our pairs of enantiomers are the mirror images. So they're the D and L conformations of each sugar. So you can have a D-glucose and an L-glucose. They're mirror images at all of their stereocenters, but not superimposable. Remember, our Fisher projections represent the open chain structures. So in the enantiomers are defined as either D or L based on the hydroxyl of the chiral carbon that's the farthest away from the carbonyl group. If it's on the right, it's a D sugar. If it's on the left, it's an L sugar. So remember, our open chain monosaccharides will equilibrate with these cyclic hemiacetals. You'll get formation of the hemiacetal spontaneously in solution. So the anomeric carbon is the carbon that, when it's in its cyclic form, the anomeric carbon is the carbon that's bound to 2 oxygens. So 2 anomers become possible when it forms the cyclic structure, because you can have the hydroxyl group either going towards the down position or the up position. We also learned that ketoses and aldoses are reducing sugars because the ketose is in equilibrium with an aldose form. So this is via an ene-diol intermediate. So remember, we could shift that bond from the ketone position to make it into the aldehyde. We also saw reactions of the hemiacetals with an alcohol group can produce the acetal, and it converts the OH group on the anomeric carbon to an OR group. So now the sugar cannot ring open, and you formed a glycosidic bond. So our glycosidic bond is also a dehydration reaction. So you're going to lose water when you combine the hemiacetal with an alcohol. So we saw a number of different monosaccharides and disaccharides in this chapter. So you should be kind of familiar with at least knowing whether the sugars are monosaccharides or disaccharides. So you should know that galactose, glucose, fructose, and mannose are all monosaccharides. And then maltose, lactose, and sucrose are the 3 major disaccharides that we talked about. And we also saw that sucrose is not a reducing sugar because there's no hemiacetal within the disaccharide. And remember, you need the hemiacetal so that you can have ring opening and you can get the aldehyde form. The aldehyde cannot be oxidized to the carboxylic acid, and so it can act as a reducing agent, or it can reduce some other molecule. We saw that chitin is a polysaccharide that's in the shells of lobsters and insects, and joints and intracellular spaces are also lubricated by polysaccharides that actually kind of look like chitin, but they're not quite as long. But they have the N-acetylglucosamine or glucosamine inside that joint material. So inside the joints, it actually acts as a cushioning agent. We also saw some other types of sugar molecules like hyaluronate and heparin and chondroitin sulfate. We also saw heparin is a polysaccharide with many ionized sulfate groups, and this can actually bind to a clotting factor and act as an anticoagulant. So it acts as a blood thinning agent. So if you were to have a heart attack or a stroke, you might be given heparin to help thin the blood to help remove the clot. So that's if a clot had formed and caused the stroke or the heart attack. So we also saw our major polysaccharides, our cellulose, which had the beta 1,4 links between the glucose monomers. And then we saw starch and glycogen with a combination of alpha 1,4 linkages in a straight chain for amylose. And then the branched form would have alpha 1,4 links and the alpha 1,6 linkages. This would be the amylopectin form of starch, which makes up about 80% of the starch molecule. And then glycogen is similar to amylopectin but has more branch points, so you can get energy faster. All right, and these are your key words, and then you are done. End of chapter 8.